go. Yeah, let's go. All right, okay. All right, okay. Return to the Mac. Get on what it is, what it does, what it is, what it is.
Aloha, I'm Joy Galloway, a student of nutrition science at the University of Hawaii at Manoa. Today I'm going to be telling you all about dietary protein, its basic structure, how our body metabolizes it, what we can use it for, and how we can obtain the right amount from our diet. So sit back, relax, grab a steak, and enjoy this short educational video. You may be wondering, what exactly is protein? Proteins are large structures made up of over 50 smaller units called amino acids that may be over 100,000 units long. Proteins are found in every cell of our body and take on various forms and functions. The basic unit of the protein is the amino acid, called such because of its amino group and acid group. There are 20 different amino acids that are used as the building blocks of the many thousands of different proteins in our body. While the common features of all amino acids are their carboxyl group and amino group, what makes them different is their side chain, unique to each specific amino acid. Of the 20 different amino acids, only 9 of these are essential in the human diet. Essential amino acids are ones that must be consumed in the diet because our bodies either cannot synthesize them at all or cannot make them in the quantities that we need. Non-essential amino acids can be made from other amino acids in a process called transamination. Transamination is transferring the amino group of one amino acid to the carbon skeleton, essentially an amino acid without its amino group, of another. This reaction uses an enzyme called an aminotransferase. The carbon skeleton that remains from the donor, in this case alpha-ketoglutarate, can be used for energy or other compounds, but I'll explain that later. There are 9 essential amino acids: phenylalanine, isoleucine, threonine, histidine, tryptophan, leucine, methionine, lysine, and valine. The essential amino acids can be memorized by the mnemonic, "Party in the house tonight! Lend me a little vodka!"
This is the moment, tonight is the night. We'll fight till it's over, so we put our hands up like the ceiling can't hold us. Like the ceiling can't hold us.
Amino acids are linked together in proteins by peptide bonds. These bonds are made between the carboxyl group and amino groups, removing water in the process. This result here is a dipeptide between glycine and alanine. When 3 amino acids are bound together, it is a tripeptide, and several amino acids results in a polypeptide. Generally, around 50 or more amino acids is a protein. These simple peptide bonds between amino acids result in the primary structure of the protein, the basic sequence. The simple linear sequence is critical because it dictates the entire structure of the protein, how it will bend, and what it will do. The secondary structure is formed as alpha helixes or beta sheets. A common example is messenger RNA. In tertiary structures, proteins coil and clump up. Here is another example of a folded protein. You'll see here that hydrogen bonds bring amino acids at different points in the linear structure together. Electrostatic bonds, disulfide bonds, and hydrophobic interactions also cause the protein to form its tertiary structure. Quaternary structures have multiple subunits, for example, hemoglobin. These complex structures are essential to the unique function of each particular protein. Now that we understand the basic chemical structure of proteins, we can talk about how our body digests protein in the food we eat. In digestion, Our body breaks down the protein we eat into individual amino acids to either be used to make energy or other products in catabolic pathways or to be reassembled for body-specific protein needs in anabolic pathways. When we eat protein, no digestion takes place in the mouth or esophagus, but there is important digestive action in the stomach. In the stomach, the parietal cells secrete hydrochloric acid, which, as you know, is highly acidic. This strong acidity denatures secondary, tertiary, and quaternary structures of the proteins by breaking their hydrogen bonds. This essentially uncoils or unfolds the long amino acid strands, inactivating their biological activity. Hydrochloric acid also has another important function in protein digestion. The chief cells of the stomach secrete an inactive enzyme precursor, referred to as a zymogen, called pepsinogen. The secretion of pepsinogen is controlled by the hormone messenger gastrin, which is released by the stomach whenever you think about food or chew. Gastrin also stimulates secretion of hydrochloric acid, and hydrochloric acid activates pepsinogen into its new active form called pepsin. Pepsin is considered an endopeptidase enzyme. It attacks internal bonds in the polypeptide in a hydrolytic action. So, already in the stomach, long proteins are being cleaved into smaller peptides. The substance in the stomach is now called chyme. Chyme is released slowly into the small intestine through the pyloric sphincter at the rate of about 1 teaspoon per minute. Now in the small intestine, the presence of polypeptides stimulates a hormone called cholecystokinin, also called CCK, to be secreted from the walls of the small intestine. CCK stimulates the pancreas to release important proenzymes that work to digest proteins. CCK also slows down the motility of the GI tract to give these proteases time to cleave the proteins. Along with CCK stimulating the pancreas, another hormone is released in the small intestine called secretin that travels to the pancreas and the circulatory system. The secretin stimulates the pancreas to secrete what we commonly call pancreatic juice. The bicarbonate in the pancreatic juice neutralizes the effects of hydrochloric acid and pepsin that came from the stomach and keeps the rest of the digestive tract from being ulcerated by such low pH. The higher pH from the bicarbonate also allows the zymogens, which are active only in certain pH ranges, to become active enzymes. The zymogens from the pancreas are trypsinogen, chymotrypsinogen, proelastase, collagenase, and procarboxyl peptidases A and B. Trypsinogen is activated into trypsin by an enzyme in the brush border of the small intestine called enteropeptidase. Trypsin then is the leader of the digestive brigade, converting the other zymogens and more trypsinogen into their active enzyme forms. Like pepsin, trypsin and chymotrypsin are endopeptidases, but the carboxyl peptidases are exopeptidases, meaning they cleave polypeptides at the carboxyl end. Other peptides are found on the brush border of the small intestine, and some of these are aminopeptidases, cleaving peptide bonds found at the amino terminus. Small peptides and free amino acids cross the membrane of the microvilli of the enterocyte of the small intestine through many different carrier transport mechanisms. Inside the cell, intracellular peptidases further break down any remaining peptide bonds into free amino acids. Amino acids are used in the small intestine cells themselves, though most enter the circulatory system and travel directly to the liver via the portal vein, but some travel on to other cells, contributing to the body's amino acid pool. You will see there are many different uses for these amazing macronutrients. There are over 100,000 different proteins in our body. Every cell needs proteins to function, and these functions are far-ranging. For simplicity, I will go over the basic roles proteins play in the body, but first, let's talk about the fates amino acids might have in the liver before they are even built up back into proteins. Oxidation for energy, conversion to glucose, and conversion to fat. For individuals consuming adequate calories and adequate amounts of carbohydrates, amino acids provide an insignificant source of ATP. However, during prolonged fasting and prolonged strenuous exercise, some amino acids can be used to make energy through aerobic metabolism in the Krebs cycle. Using isoleucine as an example, this amino acid is first deaminated in a transamination reaction. Through 6 steps, the keto acid of isoleucine is oxidized to yield acetyl-CoA and another compound called propionyl-CoA. As you can see in the citric acid cycle, acetyl-CoA enters the cycle, yielding an ATP with each turn, and ultimately 17 ATP through its electron products through the electron transport chain. You'll also see that propionyl-CoA is able to undergo a series of reactions to produce succinyl-CoA, which also enters the citric acid cycle. It is not uncommon for different amino acids to have more than one entry access point in aerobic metabolism. That being said, different amino acids enter the cycle at different points. This is an important fact for our next topic. Maintaining blood glucose levels in a certain range is a tightly regulated mechanism in a healthy body. But without a glucose supply, the body will use amino acids and convert them to glucose. The process of producing glucose from non-carbohydrate sources is called gluconeogenesis. Not all amino acids can be converted to glucose. Leucine and lysine cannot. The amino acids that can be converted to glucose, called glucogenic amino acids, may do so because they are able to be converted eventually to oxaloacetate. Oxaloacetate is able to be converted to phosphoenolpyruvate, and then 2 of these 3 carbon molecules move up the pathway in the opposite track of glycolysis to form glucose. The amino acids that cannot form glucose are called ketogenic amino acids because they can be converted to what are called ketone bodies. The brain is able to use ketone bodies as a fuel source. And remember, the brain is the tissue that cannot use any other substrates except glucose and ketones for energy. You see there are some amino acids, note our example isoleucine, that are both glucogenic and ketogenic. There's a common misconception that consuming excess proteins will have this effect. But this is not true. When we consume more protein than our body needs, we convert the excess calories into fat. Amino acids can enter the lipogenesis pathway by being converted to acetyl-CoA. Excess acetyl-CoA in the mitochondria, where the Krebs cycle is transported to the cytosol, where the enzyme acetyl-CoA carboxylase converts it to molanyl-CoA. With molanyl-CoA, through a series of many steps and the addition of 7 acetyl-CoA, the 16-carbon fatty acid palmitate is formed. From these fatty acids, the body can store them as triglycerides, the primary component of adipose tissue. The interchange between the different energy substrates in our body is remarkable. One thing I need to mention, though, is that the catabolism of amino acids produces ammonia from their deamination. Ammonia must be excreted because our bodies only tolerate certain levels of it. But the liver has a mechanism for safely disarming ammonia and converting it to a substance called urea. Urea is able to safely be excreted in the urine. When amino acids are not needed for these purposes, they are able to be used as building blocks for proteins. The process of protein synthesis is effected by our genes. Our genes and our DNA are made up of coding sequences using 4 nucleotides, which, in groups of 3s, code for specific amino acids. But inside the nucleus of the cell in these double helices, our coding is useless to us. Messenger RNA to the rescue! mRNA is produced in the nucleus of the cell through a process of transcription. All parts of this code that do not include coding for protein synthesis are removed, and the mRNA is able to exit the nucleus and enters the cytosol, where it can undergo a process called translation. In translation, the mRNA travels to the ribosome, the manufacturing site of proteins. Another strand of ribonucleotides called tRNA functions as an interpreter between the nucleic acid sequences from the mRNA and peptide sequences by picking up amino acids and matching them to the proper sequences as is coded for in mRNA to build the desired protein. There are countless functions of proteins in our bodies, and we are still learning more and more about them. I'd like to at least show you some of the major functions of proteins that are in the human body. One of the largest users of amino acids in the body are proteins for body structure, connective and muscle tissue. One example is the proteins actin and myosin in muscle tissue. Another use of proteins is for transport. An example of transport in the blood is the protein transferrin, which transports iron, which has critical roles in many metabolic processes. Another example of a transport protein is the carrier GLUT4, responsible for bringing glucose into muscle and fat cells. Speaking of GLUT4 and glucose, another important use of proteins is as messengers in the body. Insulin is a protein that is a hormone that signals GLUT4 to actually go to the membrane so that it can bring in glucose. Another messenger, which is derived from an amino acid and not a full protein, is serotonin, a neurotransmitter derived from tryptophan. Serotonin is most well known for promoting a healthy mood, but you probably didn't know that 90% of our serotonin is in our GI tract, where it helps regulate movement. Enzymes are another extremely important protein product. Enzymes enable reactions to occur in our body and help maintain homeostasis. Consider one of the enzymes discussed earlier, trypsin, which is itself a protein. And did you notice the enzyme RNA polymerase, without which we wouldn't even be able to make proteins? Enzymes work only in specific pH ranges. At this point, you shouldn't be surprised that proteins play a critical role as pH buffers, keeping cells and interstitial fluid at optimal pH. Proteins also function in immunity. One example is immunoglobulin A, which is an antibody that fights off pathogenic bacteria and viruses. An example of a single amino acid influencing immunity is a derivative of histidine called histamine, released by mast cells to produce the inflammatory response. The last role I'll mention is maintaining fluid balance in the body. The protein albumin in the blood, which, by the way, also carries fatty acids, sodium, calcium, pharmaceuticals, and other important chemicals in the blood, is a chief regulator of osmotic pressure in the blood, helping to maintain proper intercellular and intracellular fluid levels. With all these important functions amino acids and proteins have, it's now important to talk about the recommendations of dietary proteins. The adult RDA for protein is 0.8 grams per kilogram of body weight, which is also equal to 0.36 grams per pound. You may also see the RDA for protein being 46 grams for women and 56 for men, but this is simply based on the reference weight of 125 pounds for women and 154 for men. The acceptable macronutrient distribution range for protein in adults is 10 to 35% of your caloric intake. For a 2,000 calorie diet, this is 50 to 175 grams. Individuals who may require more protein than 0.8 grams per kilogram are those who are ill, athletes, growing children and adolescents, and pregnant and lactating women. Another group of people who require more total protein is vegetarians. Proteins from animal sources, such as dairy, eggs, meat, and fish, contain the essential amino acids in ratios that our bodies need. These are called complete proteins and are sometimes referred to as high-quality proteins. Non-animal sources of proteins, such as legumes, nuts, grains, and vegetables, also contain the essential amino acids, but are referred to as incomplete proteins because they are limited in one or more of these critical building blocks. What is a limiting amino acid in one food may not be in short supply in another, so vegetarians can obtain all their amino acid requirements by eating a variety of different protein sources. I hope you enjoyed learning about protein, and from Mojo and me, a warm aloha.